Welcome to part 2 of the protein structure and function chapter. We're going to now look at some of the forces that hold not only the structure together, but also the interactions that the protein is going to make. So just an example of the same protein that we looked at the beginning of the other video, where we have these beta sheets and alpha helices, which we'll discuss, and in a short sequence of adenosine poly A RNA. So there's 1, 2, 3, 4, 5 different bases here, and there are interactions that are holding this on to this poly A binding protein. So let's talk about some of those different intermolecular interactions. A few of these that we looked at in terms of the interactions that we can have covalent bonding. So here we can see a disulfide bond where we actually have electrons are being shared between two sulfur atoms that are associated with two different amino acids, but they're linked together here. And so we can kind of force the structure to be held together by these disulfide bonds. We can then also reduce those and break those apart by adding hydrogens onto those, and then they would be more polar than they were previously, maybe then interacting with more of the outside, or we can force them together in these disulfide bonds and then they become nonpolar and typically will kind of move together within or internalize themselves within a protein structure. So we have a type of covalent bond. We also have a hydrogen bonding. So we can have bonds where hydrogen is attached to a more electronegative element. And that would be nitrogen, oxygen, for example. Typically, those are the only two that you'd see. If it was a sulfur, for example, attached to a hydrogen, we could also consider that it's a weaker hydrogen bond, but usually it's oxygen or nitrogen where the hydrogen is interacting with something else. So it's only a hydrogen bond if that hydrogen is attached to a more electronegative element. And here is a hydrogen bond. It's one of the stronger types of intermolecular forces. We can have also a different type, a weaker type of bond that's very similar though, is the dipole-dipole. So if this were two polar groups coming together where a hydrogen wasn't involved in that, then that would be a dipole-dipole type of interaction. Ionic bonds are where we would see full positive and full negative charges. So not partially positive, partially negative things. It's where there's an entire full change in the charge, and where those would be creating these salt bridges, for example, or these positive and negative charges coming together. So that would be an ionic bond. That's a very strong type of bond. This is the strongest of, well, obviously the covalent bonds are the strongest bonds on here. The intermolecular forces that don't involve complete sharing of electrons, this would be the next strongest. Then hydrogen bonding, and then the weaker ones would be the dipole-dipole bond where a hydrogen isn't involved. And then finally, the weakest interactions are the van der Waals, the hydrophobic interactions. And these are where, again, it's more like lipophilic than hydrophobic. But there are some temporary induced dipoles, and we've talked about those in chemistry, but it's basically where we have this flowing of electrons around all of these atoms, and they can kind of induce a fluctuation, a wave fluctuation, and help these things stick together. So again, hydrophobic, not the best terminology. Probably lipophilic would be better, but that because really you wouldn't think about an interaction or a stabilizing force to be something where things would be moving away from each other. So it's kind of a weird way to term that, honestly. So van der Waals, London dispersion, lipophilic interactions, those would be preferred to hydrophobic. But similar idea, these kind of lipid fat kind of hydrocarbons come together, and there is a very weak interaction that does help maintain those interactions and help squeeze those together a little bit. So this would definitely be the weakest of all of these forces shown. So for this chapter, I have a worksheet. I'd like you to, on the back side of that worksheet is part 2 of some of the learning questions I have here. So I would like you to write up the carbon structure for the following 4 different peptides. So one peptide that's interacting with water, one with a lipid bilayer. So again, these should have components of 4 amino acids that might interact with a negatively charged metabolite, and then identify and draw a pair of amino acids that would interact via an ionic molecular force. So 4 different peptides, at least 4 amino acids long for each one of them. I want you to draw the whole thing out. I don't want like a little squiggly line. I don't want it to look like this. I want the whole entire, I want all the backbone. I want all the carbon, right, nitrogen, oxygen, all of them that are involved in that full chemical structure of 4 amino acid peptides. So write that out on that worksheet, and then move on to the next slide. One of the concepts I'd like to talk about in this part of the chapter is the idea that there are different levels of organization for protein structure, and it helps us have conversations about what is interacting with what intermolecular forces are happening, and where are those things happening. So the first level of the protein structure is the primary structure, and that is the description of the amino acid sequence. So it's based on that covalent backbone peptide bond that's created between the amine group and the carboxylic acid group that I talked about in the first video. So that's just based on the sequence, and that's determined by the mRNA sequence, which is determined by the DNA sequence. So that is just going to be defined based on whatever we know the genetics of our protein is. So that's going to be the basis of the primary structure, is just basically that sequence of that amino acid sequence. The secondary structure is now a backbone interaction. So it's the folding, and it's typically defined by alpha helices and beta sheets. And we'll look at those and what those mean in a little bit. It could be random coils of interactions. That is also okay. But it's really how it's defined is that we're talking about hydrogens between the amine groups and the carbonyl groups of the peptide backbone. And we'll show that to you, but it's a backbone interaction. So then that will help when we go down to the tertiary to understand the difference here. When we're looking at 3-dimensional folding based off of side chains. And again, we can use the side chains we talked about on the last slide, disulfide bonds between cysteine groups. We can have hydrogen bonds, ionic bonds, van der Waals, or London dispersion interactions, whatever you want to call those. The lipophilic interactions. So all of these different types of interactions can be happening in a tertiary structure. And the big thing here is that the tertiary, it's happening with the side chains. Secondary is happening from the backbone interactions. Tertiary is happening because of the side chains. So there is a difference between, that's the distinguishing factor between secondary and tertiary. Quaternary is when we put together multiple groups of peptides. So it's the same different types of intermolecular forces that we can have, which are all of them, but the difference is that we're adding another peptide or another protein that's separate. So it's got a separate N and a separate C-terminal region. It's kind of like if you think about a shoelace, right, forming a tertiary structure. If you take the shoelace from your other shoe and then those are interacting, that's quaternary. That's when it can be whether it's backbone or it's going to be side chain interaction. It doesn't matter. It's just whatever interactions between those two different chains and how those are connected. So this is a 3-dimensional structure of a peptide that is, you can kind of go along the chain and see where the backbone is versus some of the side chains. And so here is the N, and then we talk about the N-terminal region, NCC, NCC, NCC. We talked about the NCC as we move down, and you can kind of follow this all around through our protein, all the way through and down and around. And then I think about around here, it's a little hard because some of this is kind of folded over on itself. Must come over this way. This is the C-terminal end. So this is the N-terminal end, and this is the C-terminal end here. And we have a number of different amino acids. And you can see this NCCN. So this is the nitrogen, and it's making an interaction with this carbonyl group, that double bonded oxygen in the middle of another chain. This is actually going to be, since this is a backbone interaction, this is going to be a primary structural determination. So this, the primary structure is being held together with backbone interactions between the nitrogen and the carbonyl group, not a side chain interaction. So next we can look for one of these side chain interactions. This one's a little hard to see, where we kind of have a backbone to side chain here. So this is going to be more tertiary with a hydrogen bond between atoms of a peptide and amine group. And then we have a side chain interaction. So this is definitely a tertiary interaction structure that's happening between the two different side chains. Common secondary structures that we'll see, one of those are alpha helices, and you can see this backbone, and we can see in red here you're showing the hydrogen bonding. This is a little bit of better picture than what we were seeing before, because it's actually showing you the hydrogen that's attached to the nitrogen and how it's interacting with the red oxygen here. So oxygen and the hydrogen attachments. And this is just kind of showing you the backbone part without the hydrogens, but it's going to form this kind of helix that where the protein's going to spin around, and then those interactions are what are holding together that ribbon structure that we see in this cartoon. So different types of subunits, different types of side chains can influence and change the orientation and the tightness of that turn. You can see the difference between a left and a right-handed turn and how that's going to influence the structure and the folding. But these are basically all, again, held together by those backbone interactions with the amine hydrogen on the nitrogen, and then the carbonyl oxygen with those hydrogen bonds. Hydrogen bonds and alpha helices are really common in protein structures, and they're really important, an example of this in terms of interactions with the membrane. So typically these will self-organize, and as this protein synthesizes, it'll be synthesized into the membrane. And typically what you would expect for the types of the regions that are going to be interacting with the membrane, these amino acids in the side chains that are coming out here are typically going to be lipophilic, right? They're going to be those hydrocarbon types of side chains, where sometimes these are held together in the middle. If there is a pore or something else, they might have some other things that help to tether and tie these together. And we'll talk more about that when we get onto the tertiary structure. But these are very commonly seen within lipid bilayers. And like you saw on that previous slide, where you have multiple alpha helices kind of grouped together, typically you'll see little stripes like this you can see the green. Those are going to be hydrophilic or hydrophobic stripes, depending on where it is located. And that helps glue these together. And that's, again, we're going to more talk about, these are going to be more tertiary structures when we start to use side chains to interact things. But again, the secondary structure is this alpha helices is held together by the hydrogen bonding of the backbones, and then anything with the side chains would be more of a tertiary structure. The second type of secondary structure, again using those backbones and backbone structures, the hydrogen off the nitrogen and then the carbonyl oxygen. You can see here a couple of examples of that. This is the beta sheet, and so these are typically going to be kind of more flat. There's no kind of curve or turn in the structure. It kind of zigzag back and forth, and you can kind of see that in this cartoon representation here. And this is a one where we have the anti-parallel. So we have kind of going up this way and then it turns. Sometimes that'll be a proline, so they can have a quick hairpin turn and then it comes back and turns around again. So this is anti-parallel. There are different ways of doing this. You can have anti-parallel. You can also have parallel, where you'll have these strands will kind of wrap around in this way. Here's an example of misfolded proteins, and this is how we typically will generate, and I flipped this to the side, but you can kind of see these big strands of secondary structure, these beta sheets that form with the amyloid fibers, and this is something that you have these misfolded proteins become insoluble and stick together, and it's all backbone based. It's all using the backbone to kind of disorganize and create something stable. These are very stable, extremely resilient structures that misfold other proteins, and then obviously you could imagine if these kind of build up and extend within a cell, it's going to take up a lot of resources and cause some functional problems. In this example, I want to look at alpha keratin in hair. It's an example of a fibrous protein with alpha helical structures, and it's made up of a lot of different amino acids, including cysteine. Silk fibroin is also a fibrous protein, but consists mostly of alternating glycine and alanine, and it forms beta sheets. So if you're able to grab both ends of keratin, and you would find that it would be able to be elastic. And in contrast, fibroin is not extensible or tensile and very resistant to breaking. So I would like you in this question to explain the differences based on the amino acid sequence and the structure that describes and defines its flexibility or inflexibility in this way. The next level of protein structure is the tertiary structure, and it's going to be held by different forces, but always going to be interactions with the side chains of a protein. So those R groups are what we're looking at here. When we're talking about covalent bonds and the disulfide bonds, those are always going to be tertiary structural determinants because they are side chains. So this is a cysteine, and here you can see that sulfur. If you look at the periodic table, you could tell that there is a difference in electronegativity between the sulfur and the hydrogen. So this is a polar group, and it's an analogous group to hydroxide or an alcohol group, an OH group. And so there is going to be some, for example, hydrogen bonding that could happen between these two components. But we can also have chemical reactions where these are oxidized, where basically we lose these hydrogens, and then the electrons that are being used to hold on to the hydrogens are going to be shared between the sulfur groups instead. And so this is an oxidation reaction. Then this goes from a hydrogen bonding situation to now a covalent bonding situation. So this would be here interaction between the side chains. So you can see also down here, same thing. It could be between a similar group, so it could be one peptide. So technically this is going to be a tertiary structure, and this would be a quaternary structure because it's between two different groupings here between the peptides. So this is a tetramer of neuraminidase, and you can see that each of these different components here, this is a little cartoon kind of demonstrating these different components here of that neuraminidase and the different beta sheet groupings. Those are also kind of domains, they're called. So the different domains are defined by different colors in each of these different groups. So there are 4 different peptides, and then they have interactions between these different. So you can always see this blue and the orange seem to be where that interaction is holding these things together. These are quaternary structures, and typically it doesn't really matter how that's happening, if it's a backbone hydrogen bond or a side chain. It's just based on how many chains are involved, and that interaction is again defined as a quaternary structure. So it could be a sulfur bond, a disulfide bond, for example, or it could be a hydrogen bond in a backbone or a side chain. but that's just that interaction between different chains. At the beginning of the lecture, I talked about the poly A binding protein. Anytime that you have something that is binding a protein that's binding something else, and it could be another protein, it could be a piece of ribonucleic acid like an RNA, it could be a heme, any prosthetic group, any other type of organic molecule that is involved. This is also a quaternary structure. So here is an example of a pyruvate dehydrogenase complex, and it has 3 enzymes and 5 other proteins that form this multi-enzymatic complex. And so it is going to bind these hemes, and it's going to use those different intermolecular forces to do that. And this is, again, would be defined as quaternary structure because it's between a protein and some other component. Quaternary structures are what allow those proteins to assemble into bigger filaments and sheets and spheres and really make larger components of cells and allow them to have more ability to do unique functions. We can have dimers, trimers, quatmers, all and longer than that, right? We can have all sorts of different types of filamentous proteins, and we can create tubes and spheres and do all sorts of things. And again, typically that shape defines its function. So if you have a filament, this is going to typically be something that's going to be structural, or it's going to be allow some proteins to crawl on it, move, and generate different abilities for the cell. And tubes, I showed you that example of creating a space for proteins to fold in. So heat shock proteins and creating areas and regions where different things can happen in a safe space and a space where different external stimuli are not influencing the folding. So just the overview again, the primary structure is just the sequence, the order of from the N terminus to the C terminus, the amino acid order and sequence. Secondary, we're talking about typically alpha helices and beta sheets, but really any interaction between the backbone, that a carbonyl oxygen and the amide hydrogen, for example. So those interactions between the backbone. When we go to the tertiary structure, we're talking about the R groups, the side chain interactions, and we usually, and it doesn't matter really where those are located as long as they're happening with the side chain groups. And finally, the quaternary, where we have multiple peptide domains. So that's kind of the overview. You should be able to distinguish and identify, differentiate these different types of structural determinations and levels. So far, we've been talking a lot about the structure, and the structure is really important. Sometimes unstructured regions still have an important purpose, and it really depends on when we're looking at it, right? So if you're looking at a protein that's kind of just looks like nothing, and then it interacts with something else, then it has the structure. There are things that can determine the function, and the structure might not happen until it interacts with that. So if it's binding, it might that flexibility initially can allow it to increase encounters for those domains that they're trying to interact and bind to. So this region might act like this until it finds its thing of interest, and then it creates the structure that's important. This might not be what it looks like in its active state. The same with things that are tethered. Maybe this needs to be activated or something has to happen, but it's obviously close by. So then once something activates this protein, then it can bind maybe into now a new structure that is active. You can see this is an example. It looks like spaghetti with these different domains. And then if we have an interacting protein or something, tethers it and allows it to interact and changes the structure, we can then allows this to function properly. So it gives it some flexibility and ability to kind of bind to things and then react afterwards. So unstructured regions often still can have functions. We might just not know what those structures are supposed to be because we don't necessarily know what it's binding to yet. So sometimes it's important to find out what it's interacting, what it's doing, in order to figure out what the structure might be. In the previous video, I discussed some of the different types of proteins based on what they do. And they're also a different way to divide proteins in kind of what they look like and how how their regions kind of behave. And so here, one of the big kind of proteins are fibrous proteins. So they usually have extensive regions of secondary structures, so those alpha helices or beta sheets. They're really highly ordered, repetitive structures. And some examples of this would be fibroin proteins in the skin, keratin of your hair and nails, and collagen found in tendons and also in skin, elastins found in ligaments and blood vessels. So these are really highly organized. They're just kind of these structural components of some of our cells and extracellular components as well. So that's one, the fibrous protein. And we also have the globular proteins. And most proteins are actually globular proteins. They're folded into these compact structures. They're not elongated. They're not always really regular, right? You can see we have some alpha helices and some beta sheets, and they're kind of laying on top of each other in various different ways. These types of globular proteins have their own unique tertiary structure, and most enzymes, for example, are also globular proteins. They also typically have, like you can see, the multiple different secondary structures coming together in that tertiary state. So examples of, here are some more globular proteins. You can, tobacco mosaic virus coat protein, for example, would be globular. Here we can have all these beta sheets coming together in various organization. Here's hexokinase domain 2. You can kind of see this interesting structure as well. So bringing together, it can be alpha helices, beta sheets, mixture of both, but it's not a repetitive systematic structure all of the time. One thing I also mentioned is a lot of times there'll be breakups of different domains. So this is V2 domain, and this is hexokinase domain 2. And this is to say that you can kind of see this is cut off. This protein goes on further. There might be 2 or 3 different domains, and it might be one strand. It might be one coding sequence. So it might be, you could have a protein that might have 3, 4, 5 different domains. And what we say when we say domains is these things kind of glob onto each other and interact with each other. And a domain usually has some functional purpose. So it might, if hexokinase might be an enzyme that does multiple things, maybe this part of it binds to one component of its substrate, and then the other thing helps to enzymatically cut it in half or glue it together or interact with something else. And so usually these domains will have different functions within even one protein. So if you see domain, that typically tells us that there are probably maybe there's a V1 domain and a V2 domain, and there's maybe some other domain. That image that I showed you at the beginning of this lecture, that was the RNA binding domain. So there are other domains outside of that protein. That one just binds the RNA, and there are other domains that do other things, maybe interact with other proteins or interact with other RNA groupings. So just again, to kind of reiterate that point, we will typically, domain is a discrete, locally folded unit of tertiary structure. So has a specific function, usually between 50 and 350 amino acids, and they're packed together, and they share this secondary, tertiary structural region, and again, usually having multiple different functions. Since we're talking about proteins and we've spoken a lot about different types of assays that involve proteins, I'd like to spend a little bit of time discussing the antibody molecule, just so we can be clear on what this is. So this is typically multiple subunits that are involved in this. We'll have a heavy chain, which is a larger, longer peptide or protein, and a light chain. So the antibody together itself is held together by quaternary structural bonds, right? So we can have interactions between these heavy and light chains, and between the heavy chains as well. I mentioned different types of assays where we look at attaching antibody to a protein of interest, and then we use immunocytochemistry. So there might be something that's a secondary antibody attached to that as well. So we can create secondary antibodies by just taking this heavy chain domain, just this part of it, and injecting that into our animal of interest, and then this will be recognized by the other animal, and then this is what the antibody would bind to. So let's say this whole thing was a mouse antibody that was specific for our protein of interest, and then if we cut this out and injected this part into a mouse, then it would create, or into, sorry, a donkey, for example, it would create antibodies to just this region, which isn't involved in the binding region. So these are the binding domain sites. So if I have another antibody that's binding to this, it wouldn't really influence its ability to attach to our protein of interest. So this is how we can use this information about domains to make different assays and understand the biochemistry that's happening in a system. If you want to learn more about this process, I would recommend taking immunology with Dr. Smith. But just the long story short, we can use some recombination of the sequencing that we have for our different light and heavy chains, and we can recombine these in various different ways to make different antigen binding sites, so that the different recombination will give us those different domains, will provide different structures that can receive different types of antigens, and that's going to give us our immune system essentially. So long story short, those mutations can change what's happening, or not mutations, those recombinations can change what the structure, this domain, looks like in the end. So here's an example of just using antibodies and how it can be used to understand different and similar proteins and the power of antibodies for this. So GABA A transporter 1, GABA A transporter 2, and GABA A transporter 3 are maybe three different transporters that we're interested in studying and understanding where it's located, what it does. So GABA transporters actually move the neurotransmitter GABA. Once it's in the extracellular space, it helps to recycle that back into the neuron or the astrocytes in the brain. So if we have these three different transporters and we have antibodies that bind to the antigens, then we can, which might be GAT1, GAT2, or GAT3, then we can figure out where we might have this localized. So under normal conditions, we're going to look at the tissue is stained. If we just take the antibody and stain the brain, it's everywhere in the brain section. So we know that this antibody binds. There are proteins in here that this is recognizing. So in a normal brain tissue, yes, it's either GAT1, 2, or 3 in this tissue. Next, I want to see, okay, so I know that there are GAT proteins in the brain. That's great. We already knew that. But can we distinguish between GAT1, 2, and 3? So what I'm going to do is I'm going to take my antibody and I'm going to preload it. I'm going to put this in here, this antibody, in here with one of these 3, and I know I have 6 here, but we have GAT1, 2, and 3. I'm going to test these 3 on the bottom. And this, I want to see if this, for example, is GAT1. I want to see if GAT1 keeps it from binding here. So what happens is, no, okay, sorry about the white circles, but you can see this is kind of dark, black, like blackish. This also is. So even though we put this in here, it didn't have an influence on the ability of this antibody to bind and stain in here. So this preloading didn't do anything, and it didn't influence the staining. So we know the antibody is not bonding to GAT1, for example. Let's try GAT2. Okay, so we preload it, we put these in, we incubate them together, we kind of mix them up, and then we add them onto our brain slice. Oh, we still get staining here. So, okay, GAT1, no. GAT2, no. How about GAT3? What happens if we put this in with our antibody, we preload it, and then we see that it doesn't stain? So this we know now, we can tell that this antibody is very specific for GAT3. And then GAT3 is what's giving us this stain. So this is our negative control, and here are some positive controls. So this antibody is specific for a GAT3, and that's kind of interesting, right? Because even though those structures I showed you back here might look very, very similar, there is enough of a difference between these 3 proteins that had very similar structures and domains and sequences, there was enough of a difference to generate an antibody that was unique to that specific protein. So in the first chapter, I also gave a quick overview of where how antibodies are made, and this is just another just kind of dive into that in a little bit more detail. Again, way more detail. You can take immunology with Dr. Smith, but the general idea is that you have these beta cells or beta lymphocytes. Lymphocytes are some of those cells that are in our blood, kind of moving around and checking different functions in our body. So they're typically bone marrow derived in the blood, and these resting beta cells have these membrane-bound antibodies, so they act as little receptors. And when the antigen binds the receptors, then there's a stimulation. So there's a signaling transduction pathway that's activated throughout the cell, and then it is stimulated to divide into multiple different cells and then secrete some of these antibodies in not only membrane-bound, but just into the soluble form, into the blood. Antibodies are helpful for our functioning as living creatures because they help to protect us from different foreign molecules that might invade and cause other problems. Viruses that try to attack and hijack our DNA and RNA systems, protein synthesizing systems, and bacteria as well. So there are releasing different antibodies help to cross-link and aggregate those things so that different types of cells can come, phagocytic cells can come in and digest them, or they can get special proteins in the blood that will end up coming and killing coated bacteria or viruses to eliminate them from our systems. Typically, you'll see that you get different titers or injections over time because the amount of antibody or in the blood is increased with different types, different injection time points. And this would be where you would generate polyclonal antibodies by injecting and boosting over time to get the maximum amount of antibody. And they typically get better over time to stimulate those different B cells. A really common use for antibodies is immunoprecipitation. So if these are, and again, this isn't an amino acid, these are protein A, B, C, D, E, F, whatever. So the proteins are numbered, lettered, I guess. And so here we have if we have a mixture of proteins or things that we're looking at, you have a specific antibody, anti-A antibody, it's going to bind all of those. And if you have really complex mixtures and you want to just look for something like GABA A transporter, for example, you can grab these and pull these down and then look and see. Sometimes, depending on how you do it, you can also see if is attached to other things, D, O, F, right? So you can see what other things that A interacts with as well. So that's typically used immunoprecipitation for identifying other proteins or other things that this might interact with. A lot of times this is used in columns in chromatography as well, where it's coated, where beads are coated with the antibody so that you can purify out A from these other more complex mixtures. And so this is where you would flow the mixture through and you get all of these other things. A is attached, and then you would use maybe some type of higher salt solution or some other method for eluting off your protein of interest. So different ways of collecting, purifying, enriching your sample of interest. Antibodies are really helpful for that, in addition to the example where we're looking at staining and looking at identifying where things are bound or found within the cell. In chapter 1, I also spoke about hybridoma banks, where we take those B cells and then we introduce them and hybridize them with cancerous cells, and then we create this immortalized cell line that's secreting and making all of these antibodies. So this is indefinitely making these monoclonal antibodies. So we take that beta cell that's been activated, and then it's kind of activated and immortalized. So it never stops dividing, never stops secreting the antibody out of interest. And this part is necessary because the beta cells don't live forever. They have a finite number of cell divisions before they would senesce or stop growing. So this immortalization helps to make sure that you never run out of the antibody that you might want to use for your research studies. And as I mentioned before, that secondary, usually you use your primary antibody is the primary. It's that thing that you're interested in, whatever, if it's a protein or a bacteria of interest, that's your primary, what's attached to your antigen of interest. And the secondary one, remember I talked about the heavy chain, that component, that's where you would take that and get the secondary antibody that might be like a goat anti-mouse. So that goat antibody is binding to the heavy chain of the mouse antibody. And that is usually where we would label these things, either with gold labeled particles. If we're doing electron microscopy, you can have fluorescent tags, for example, if you're looking at immunocytochemistry, or some type of fluorescent or light emitting tag that you can use for a Western blot. So there are a lot of cool things that we can do with antibodies. And that's why I want to take a little more time to kind of walk through some of these ideas and concepts that are commonly used in biochemical assays. All right, the final learning check, and what I would like you to think about is what kind of, based on what I was speaking to you about, what kind of things can we use antibodies to detect? So list all of those that you can think of, and then what macromolecules might we not be able to detect with antibodies? So all of the things you can detect and things you think maybe we wouldn't be able to detect with an antibody. So maybe don't list all of the things that we can't detect, but at least maybe list 2 or 3 that you think we might not be able to detect with an antibody. So this is the last learning check, and this concludes the Chapter 3 summary on protein folding. And so we'll be getting into this fold-it game that is going to help us in this case study that's involving protein folding, protein structure.